Hello friends, this is Dr. Ranjini P, Assistant Professor in Biotechnology. Welcome to e-learning platform. Today we learn about the structure of chromosomes. All cells have capability to give rise to new cells, and the encoded information in a living cell is passed from one generation to another. The information encoding material is the genetic or hereditary material of the cell. Bacterial genetic material. The prokaryotic, that is bacterial genetic material, is usually concentrated in a specific clear region of the cytoplasm called nucleoid. Bacteria is having primitive nucleus is called nucleoid, and the genetic material will be in contact with the cytoplasm called nucleoid or incipient nucleus without nuclear membrane. The bacterial chromosome is a single circular double-stranded DNA molecule, mostly attached to the plasma membrane at one point. The bacterial genetic material does not contain any histone protein. This is very important. For example, in E. coli, Escherichia coli, DNA is a circular molecule having 4.6 million base pairs in length, containing 4,288 annotated protein coding genes. Means in 4.6 million base pairs, 4,288 annotated protein coding genes are present, and they are organized into 2,584 operons, 7 ribosomal rRNA operons, and 86 transfer rRNA genes are present. Besides the bacterial chromosomal DNA, many bacteria may also carry extra chromosomal genetic element in the form of small, circular and closed DNA molecules called plasmids. Apart from the chromosomal genetic material in the bacterial cytoplasm, we will find the extra chromosomal DNA that is autonomously replicating independent of the chromosomal DNA, and it is circular, and it is called as the plasmid. The plasmids generally remain floated in the cytoplasm, and they bear different genes based on their presence or based on their function. Some of the different types of plasmids are F plasmids, R plasmids, virulent plasmids and metabolic plasmids. You will be learning about these plasmids in detail in your higher semesters. This is the diagrammatic representation of the bacterial DNA. It is primitive nucleus having the genetic material attached at one end to the plasma membrane, and it is in direct contact with the cytoplasm. And it is not associated with any histone molecule. And apart from the chromosomal DNA, we also have extra chromosomal DNA which is circular and it is autonomously replicating called plasmid DNA. This is TM image of plasmid DNA, how it looks. This is double-stranded and circular DNA. Coming to the virus genetic material, the chromosome material is made of either DNA or RNA, which adapts to different structures. So The viruses, they have either DNA as a genetic material or RNA as the genetic material. When it is packaged, it is circular inside the virus particle or virus coat protein. Coming to the eukaryotic genetic material, a eukaryotic cell has genetic material in the form of genomic DNA enclosed in the nucleus. This nucleus is membrane bound organelle and it's a true nucleus. It's not like our bacterial nucleus. The bacterial nucleus is incipient nucleus where it has the genetic material directly floating in the cytoplasm. But here the nucleus is membrane bound and it is not in contact with the cytoplasm. The genes or the hereditary units are located on the chromosomes. Genes are located on the chromosomes which exist as chromatin network. In the form of chromatin network, it is located inside the nucleus in the non-dividing cells or in the cells which are at interface. Now we'll move on to the structure of the chromosome. The German biologist Walter Fleming in early 1880s revealed that during cell division, nuclear material organized themselves into visible thread-like structures which were named as chromosomes, which stains deep with basic dyes. These chromosomes, they appear as visible thread-like structure during the cell division and they take up the stain, especially the basic stain or the basic dye. The term chromosome was coined by Waldeyer in 1888. Chrome refers to colored and soma is body. Hence, they mean colored bodies as they take up the color or the dye and can be defined as higher order organized arrangement of DNA and proteins. It contains many genes or the hereditary units. Genes are otherwise called as hereditary units and regulatory elements which regulate many metabolic reactions or the many processes inside the cell and other nucleotide sequences. Chromosomes also contain DNA-bound proteins which serve in the packaging the DNA and control its functions. Chromosome also contain DNA-bound proteins which helps in packaging the DNA and controlling its functions. Benden and Boveri in 1887 reported that the number of chromosomes in each species is constant. The number of chromosomes in a species is constant. Arabidopsis thaliana has 10 chromosome and maize has 20 chromosome and human being has 46 chromosome and donkey has 62 chromosome and goldfish is having 100 to 104 chromosomes. So this chromosome number is species specific. So this table represents the name of the organism and their chromosome number. W.S. Sutton and T. Boveri in 1902 suggested that chromosomes are physical structures which acted as messengers of heredity, Chromosomes are tightly coiled DNA around basic histone proteins which help in the tight packing of DNA. In eukaryotes, we'll find this histone protein, but they are absent in case of prokaryotes. In eukaryotes, to to fit the entire length of the DNA in nucleus, it undergoes condensation. And the degree to which DNA is condensed is expressed as packing ratio. The degree of condensation of the DNA in the nucleus is expressed in packing ratio. Now, what is packing ratio? It is the length of the DNA divided by length to which it is packaged into chromatin along with the proteins. It is the length of the DNA divided by the length to which the DNA is packaged into chromatin along with the proteins is called packing ratio. So this is just the diagrammatic representation of the chromosome. This is unduplicated chromosome, and these two are duplicated chromosome. One is derived from the paternal side and another one is derived from the maternal side. And the chromosome, it is having the constricted region that is called as the centromere, and you'll find the different intensity of the dye all over its surface after it is stained because of the variation in the packaging or the condensation of the DNA all along the length of the chromosome. You'll be learning about the detailed structure of the chromosome in the coming video. This is regarding how to calculate the packing ratio. The shortest human chromosome contains 4.6 into 107 base pair of DNA. This is equivalent to 14,000 micrometer of extended DNA. In its most condensed state, during mitosis, the chromosome is about 2 micrometer length. Shortest chromosome length is 2 micrometer. It contains 4.6 into 104 base pair, that is equivalent to 14,000 micrometer. Now, how will we calculate the packing ratio? It is 14,000 micrometer divided by 2 micrometer. So during mitosis, the chromosome will be having the length of 2 micrometer. So in this condensed state, you have 14,000 micrometer length of the DNA. So 14,000 micrometer length of the DNA divided by the actual length of the chromosome, that will give you the packing ratio. The DNA is packaged stepwise into the higher order chromatin structure, and this is known as hierarchies of chromosomal organization. Now we learn about little more about chromosome number. There are normally 2 copies of each chromosome present in every somatic cell. 2 copies means 1 copy derived from the paternal side, the male parent side, and another copy is derived from the maternal side or the female parent side. The number of unique chromosomes, small n, in such cell is known as its haploid number, and the total number of chromosomes, 2n, it is represented by 2n, is its diploid number. Haploid and diploid. Haploid represents the single set of chromosome, whereas diploid represent both the set, 2 copies of the chromosome. The suffix ploid refers to chromosome sets. Haploid set of chromosome is also known as the genome. They will ask you in the short notes, define genome. What is genome? It's the haploid set of chromosome of an organism in the nucleus. Structurally, eukaryotes possesses large linear chromosomes, unlike prokaryotes which have circular chromosome. Eukaryotes have large linear chromosomes, whereas prokaryotes have circular chromosomes. In eukaryotes, other than the nucleus chromosome or the nuclear material, the chromosomes are present even in the organelle like mitochondria and the chloroplast, or the DNA is present in the mitochondria and chloroplast too. The number of chromosomes in each somatic cell is same for all the members of a given species. So the chromosome number is species specific. The organism with lowest number of chromosome is the Eukaryotic, which has only 2 chromosomes in the somatic cell. That means its diploid set will be equivalent to 2, only 2 chromosomes. In a diploid cell, there are 2 kinds of chromosome. One is autosomes and another one is sex chromosomes or allosomes. In human being, there are 23 pairs of homologous chromosome. 23 pairs means the diploid number will become 46. In human female, we have 44 non-sex chromosomes are called autosomes and 1 pair of homomorphic sex chromosome given the designation XX. In female, we have 44 autosomes and 1 pair of homomorphic sex chromosome designated as XX, 44 plus XX in female. But in human male, we have 44 autosomes and 1 pair of heteromorphic sex chromosome. In case of female, we have homomorphic X chromosome because we have 2 X chromosome. But in case of male, we have heteromorphic allosomes or sex chromosome. One is X and another one is Y chromosome. So this is regarding the just introduction towards the chromosome. We will be learning about the detailed structure of the chromosome in the coming video. I hope you understood the today's topic. Thank you.